Welcome everybody. I am Dr. Manar lecturer of medical parasitology. Today I would like to talk about family Ceratopogonidae and Psychodidae of Nematocera. This lecture will help you to know taxonomy, morphology, biology, life cycle, medical importance, prevention and control of family Ceratopogonidae and Psychodidae of Nematocera. Let's start with taxonomy. Phylum Arthropoda, subphylum Mandibulata, class Insecta, order Dioptera, suborder Orthorhyncha, which has two divisions. Nematocera, which contains family Culicoides, Similidae, Ceratopogonidae, and Psychodidae, and Brachycera, which contains family Tabanidae. Let's start with family Ceratopogonidae, Culicoid species. Many species of culicoids never attack man, but certain ones constitute a serious economic problem by their annoyance to campers, swimmers, and fishermen. In addition to the annoyance factor, these blood-sucking midges have known rules in the transmission of human and animal pathogens, with at least 5 elements involved, as Onchocerca, several protozoan and viral diseases of domestic animals, poultry and waterfowl, and possibly man are related to Ceratopogonidae transmission. Let's start with biology and life cycle of culicoids. In most culicoid species, the life cycle is poorly understood. During the summer months, the development from egg to adult occurs in 2 to 6 weeks, and some larvae and pupae overwinter in protected breeding places and continue development in warmer weather. Culicoid species undergo complete metamorphosis, which give larvae and pupae and adult. Adult culicoids usually live for 20 days to more than 90 days, depending on ambient condition. The adult fly and copulate in swarms. The mean distance for female flight is 2 kilometers, less than half of that distance for males. However, these species can exploit wind to increase their dispersal. Eggs can be cigar, banana, or sausage shaped and 0.25 mm long. They are white when first laid, but later turn brown or black. The eggs are laid on moist soil and can't withstand desiccation. Some species can lay up to 450 eggs per batch and as many as 7 batches in a lifetime. The number of eggs produced varies among species and size of blood meal. Eggs typically hatch within 2 to 10 days of being laid. Time to hatch is dependent on the species and temperatures. And larvae are worm-like, creamy white, and 2 to 5 mm long. Larvae develop 4 nesters. The first one possesses a functional spine-bearing proleg. Larvae require water, air, and food, and not require aquatic or terrestrial. Larvae can't develop without moisture. Larvae are present in and around salt marsh and mangrove swamps, on shores, of streams and ponds, and in muddy substrates. They feed on small organisms, and most species can't exist more than a few inches below the air-water interface. In tropics, the larval habitat of many species is in rotting fruit and other water-holding plants. And other habitats include tree holes and slime-covered bark, while some larvae can develop in wet, manure-contaminated area. They don't develop inside the animal. The larvae also don't develop inside humans or other animals. The pupa stage typically lasts 2 to 3 days. Its color can be pale yellow to light brown to dark brown. They are 2 to 5 millimeters in length, with an unsegmented cephalothorax that has a pair of respiratory horns that may bear spines or wrinkles. During this stage, the insect possesses a spiny integument, which can be used to identify to species level. And the adults are gray and about 3.17 millimeters long. The two wings possess dense hairs and give rise to pigmentation patterns, which are used to identify species. The mouth parts are well developed with cutting teeth on elongated mandibles in the proboscis of blood-sucking female, but not in males. Male culicoides typically emerge before the females and are ready to mate when the female emerges from the pupal stage. Mating occurs in flight when females fly into swarms of males and the insects are oriented end to end with the ventral parts of the genitalia in contact. And some species mate without swarming. Instead, the males go to host the female, is likely to feed on blood. Mating occurs when she finishes feeding. Both males and females feed on nectar, but the females require blood for their eggs to mature. This picture showing culicoid species, showing mottled wings and mouth parts. A on the left side, adult female, and B on the right side is mouth part, which consists. After morphology and the life cycle, let's talk about medical importance of culicoid species. Number 1, 3 non-pathogenic filarial nematodes are transmitted to humans by Ceratopogonidae, Manzanella bristans and Manzanella streptocerca in Africa, and Manzanella ozardi in South and Central America. Onchocerca cervicalis of horses and Onchocerca gibsoni of cattle are transmitted by culicoid species, as are other filaria of domestics and wild animals. Number 2, hepatocystis species is in monkeys and other arboreal mammals, some species of Hemoproteus in birds, and various species of Leucocytozoan are transmitted by Ceratopogonidae. 3. Blood-dwelling protozoan parasites also use culicoids species as vectors. Number 4. Orbiviruses, the viral etiological agent of blue tongue, is spread by culicoids in North America and by other species of culicoids in Africa. Blue tongue is a hemorrhagic disease of ruminants, including sheep. It causes some mortality, but infected animals suffer mainly from loss of weight, loss of wool, and reduced breeding. Number 5. Ceratopogonidae may also transmit encephalitis viruses, bovine fever, and African horse sickness. After family Ceratopogonidae, we will talk about family Psychodidae. Family Psychodidae has two subfamilies are of medical importance. Subfamily Psychodinae contain moth flies which are of only slight parasitological significance. Subfamily Phlebotominae consists of sandflies of great importance in many parts of the world. About biology and life cycle, in subfamily Psychodinae, the body and ovoid wings are densely covered with hairs. The roof-like position of the wings when at rest suggests the appearance of a tiny moth, hence their common name moth fly. And these flies breed in substrates rich in organic decomposition. Psychodinae alternata, the trickling filter fly, is often found in incredible numbers in sewage disposal plants. It and other species are common and drains, where larvae develop in gelatinous lining that commonly accumulate in babies. Emerging adults may be so numerous as to constitute a nuisance to householders. In addition, larvae have been reported in pseudomyiasis, and this species can be involved in mechanical transmission of nematodes parasitizing livestock. Subfamily Phlebotominae. Sandflies go through complete metamorphosis, progressing from egg to larva, pupa, and adult in 5 to 11 weeks, depending upon the temperature and the larval food quality. And the eggs are elongated, oval, dark brown in color. The eggs are 0.31 millimeter long and 0.1 millimeter wide. The average duration of the egg stage range between 6 and 13 days. The first instars are pale cream in color with a body length measuring 0.4 to 0.68 millimeter, while the color remains similar to the first instar. The second and the third one are larger in size, and the fourth measures 2.2 to 2.5 millimeter. The larvae feed in organic debris, and the average duration of the 4 instars range from 21 to 51 days. Pupae are optic, measuring 2.6 millimeter in length and look much like a butterfly crystal. They are glued to the substrate by the fourth instar. The pupa generally require 10 days. Adults are small, silver brownish, long-legged flies with narrow bodies. The dense hairy wings are 3 mm long and are held in a vertical V over the body. They are nocturnal active, with weak direct flight, typically not dispersing more than 0.5 km unless transported by wind. Adults shelter during the day in dark, humid places such as trees holes or under rocks, and sexual dimorphism is marked between the male and female flies. Males have external terminalia with a relatively small and slender abdomen compared to the female. Mandibles are absent in the males. The females require vertebrate blood for maturation of the follicles. The first oviposition begins between 4 and 8 days after blood ingestion, and the males and females feed on nectar and other plant juices, but males don't take a blood meal. Eggs are laid in small batches in humid habitats, rich in organic matter that provide food for larvae. Let's talk about medical importance. Sandflies are good vectors of disease, which is surprising considering their weakness and fragility. They transmit leishmaniasis, including those of non-human vertebrates, Bartonellosis, and some viral diseases. And the studies show that salivary secretions contribute far more than previous suspected to Phlebotominae vector potential and leishmanial pathogenesis. Saliva alters the course of leishmania infection through injection of a vasodilator protein, maxadilin, modulation of cytokine production by antigen-producing cells, and inhibition of complement activation. Saliva proteins also have been used as vaccines in mice, and immunization with saliva results in protection or severity of infection. The bacterium Bartonella bacilliformis causes a disease known as Oroya fever, being transmitted by Lotzomia. Oroya fever is a sometimes fatal visceral form of the disease accompanied by bone, joint, and muscle pains, anemia, and jaundice. Sandfly fever is transmitted by Phlebotomus babatesi and other flies in much of the Old World, also known as babatesi fever and 3-day fever. It occurs in the Mediterranean region, southern China, and India. And sandfly fever is a non-fatal, febrile, viral disease of short duration, but with a long convalescence period. Sandflies acquire the virus when they feed, but because males don't feed on blood and also are sometimes infected, it seems that transovarial transmission occurs. Finally, These are references if you want to have a look. Hoping that lecture was useful for you all. Thanks and goodbye.